Okay, can everybody see my screen? Okay.
Yes.
Hi everyone, I'm Jack. Welcome to your last homework 6, and this is lab 10. A few announcements before we dive into the homework. So homework 6 is due not on Sunday, but on the last day of this course. So it's Tuesday, April 27, midnight. So you have, I think, 2 more days to work on the homework. And the office hours in the next 2 weeks will be held on regular hours. So Friday, April 9th and April 16th from 4 to 5:30 p.m. and Saturday, April 10th and April 17th from 10 a.m. to 12 p.m. And for this homework, I know people are really from different backgrounds, and some people want more specific help on coding. Some people have difficulty in understanding the main idea and approaches to a question. So I try to take a balanced way. So in this lab, I will mainly focus on the main idea for solving each problem. And I will, of course, include a few codes if that's necessary, and some sample nodes that you'll actually need to use to finish the homework will be provided in a separate file. So a bit of background first. For homework 6, we will be focusing on glioblastoma, and it is a type of aggressive brain cancer, and it starts in astrocytes, um, which is a type of cell that supports your nerve. And where you will be asked to do some simple TCGA exploration. So uh, across 36 cancer types. So for question 1, you are asked to simply just count the number of samples that satisfy a series of criteria. The first criterion is that you need to subset samples corresponding to glioblastoma in TCGA. So it's really easy. Just go to this website and you will see the page here and click on exploration and then cases. So you will find tab for primary site, project, and disease type. And you should click on the correct option that uh, gets you glioblastoma data in TCGA. So for example, like for primary sign, maybe you want to click on a check brain. Uh, and I think another several other criteria should be like I think gender, vital status, and age. And it's similar idea. You just click on the clinical tab and check male, and I think in vital status you should check alive, and then under age here you just type in the correct age, the range of age the question asks you to subset, and then at this place it will show you the number of samples that satisfy these few criteria. That's really simple. Broad file host. So you need to first install the relevant package file browser. It facilitates efficient download of processed TCGA data. So you need to use the DevTools, but if you haven't installed that yet, you should first use install packages DevTools and then install from GitHub. This is the vignette for file browser, and feel free to explore it. So question two asks you to get process data of glioblastoma from file host. The command you will be really using is just sample.clinical cohort equal to a cancer type. Here I use BRCA as an example, but you will need to find the correct abbreviation. This is abbreviation for a cancer type. You need to find the correct abbreviation for glioblastoma yourself. So the command you will be using should be metadata cohort. You will print out a data frame with two columns. One column is the abbreviation for cancer type. Another column is the detailed description. So you need to read this column yourself and find the correct abbreviation and put it here. It's kind of weird that in this way you just get actually 150 patients, because we are allowed to really just download 150 patients at a time. So to get all the patients, maybe thousands of patients, you actually need to do this in a for loop, like this one shown in the screenshot. So don't worry, I have already provided this chunk of code in the sample code file. You can just run this code, copy this code and run yourself. The only thing you need to change is this abbreviation for glioblastoma. So I won't explain the details of this chunk of code, but it's just a for loop that extract 150 patients each time and store it as an element of a list. And then we use this do call rbind command to row bind every element of that list to generate the final data frame. So for question two, you will get a data frame in the end with patient in rows and a bunch of variable measures in columns. And it asks you to calculate the average age at diagnosis. So you need to find a column that corresponds to the average diagnosed age and just calculate the mean. So the calculation part should be really easy. It's kind of weird. My internet is kind of slow. Okay, stars. So for part two, we will focus on differential expression and differential methylation of genes using microarray data. So this is an overview of the microarray mechanism. So microarray is kind of an old way of measuring gene expression before the rapid development and adoption of RNA sequencing. So the basic idea is shown here. Suppose you have cell cultures from different conditions. Say you have normal cell cultures versus cancer cell cultures. And you obtain their mRNA and cDNA. And you label the cDNA from different conditions with different fluorescent probes. So in this case, for example, we label cancer cDNA with red probes and normal cDNA with green probes. And the idea to measure gene expression is through hybridization on a microarray or a chip, where each slot here, each probe contains a gene sequence. So if your CDNA, for example, cancer cells expresses a certain gene, and the corresponding cDNA will hybridize with that gene probe on the chip, and it will show you a color. In this case, it's red. So when you see a slot is red, that means that gene is specific to cancer cells. And similar idea, similarly, like if a slot is green, and you will know that the gene is specific to normal cells. And if it's yellow, which is just a combination between blue and red, and you know that this gene is present in both cancer cells and normal cells. So on the right part, it's the data processing workflow for microarray data. So from your hybridized gene chip, you do a scan and you get a DAT file that contains image information. And you process the image to get a CEL file, which contains the intensity value of each probe. So the color intensity value for each probe on the chip. But you will also need a CDF library file that tells you which probe belongs to which gene. And then you can get a conventional expression matrix, like with patient in the columns and genes in the rows for downstream analysis. So for part two, question one, we will provide you with an already processed microarray data that contains 60 tumor samples and 10 normal samples. The expression matrix is already log2transform, and if you read it in, it will look like this. So patients are in columns, and tumor samples starts with TCGA, and normal samples I think start with just the string normal. And genes are in the rows, and each cell contains the log2 expression value. So question one asks you to do a k-means clustering using separating this 70 samples into 3 clusters to determine whether different glioblastoma subsites exist. So if you doing this clustering, and if you observe that the 10 normal samples always group to one cluster, and and among the 60 tumor samples, some of them always group to one cluster, and the others group to another cluster, and this is indication that different glioblastoma subtypes exist, and you will be asked to use all genes and top 200 variable genes to perform k-means clustering. So I won't talk about how exactly we should do k-means clustering because you already did that in homework 2. Just be careful that for all genes, you use all the rows or genes in the rows. And for top 200 variable, top 2000 variable genes, you need to calculate the variance of each gene yourself, rank them, and choose the top 2000 variable genes to perform k-means clustering. And for this kind of large data set, I would recommend you use the following parameters, and star equal to 10 and the maximum iteration number being 100. And starting from part 2, question 2, you will be asked to perform differential expression using Lemma. So Lemma is just like the ESIG2, a bioinformatic tool that allows you to figure out gene differential expression across different conditions. And for Lemma, the idea for Lemma is really simple. Lemma just fits a linear model for each gene. So it's linear regression showing in the formula here. So Yij here represents the expression value for gene J in sample I. Well, beta 0 is the baseline expression for gene J, and Xi is the covariate for sample I. So for example, if you want to know if the genes that differentially express comparing tumor samples to normal samples, Xi should be an indicator variable indicating whether this sample is tumor or normal, so 1 or 0. And the beta J is the difference of expression in this gene comparing tumor to the normal samples. And as you can see, beta J will be the magnitude of expression change comparing two conditions, while the significance of this coefficient will be the significance for differential gene expression. And you use p-values of beta J to obtain to call differentially expressed genes. And of course, since we are fitting this model for a bunch of genes, maybe tens of thousands of genes, and multiple testing correction will be needed. Okay, I will answer your question at the end of this section. So specifically for this question, we're asking about what are the differentially expressed genes comparing the two tumor subtypes. So you need to first create the variables to indicate whether a sample belongs to subtype 2 or subtype 1. So say we use subtype 1 as the reference group, and the gene expression is the mean log2 expression will be for gene is beta 0. And in the reference subtype. And in subtype 2, which is sort of like the test subtype, you will have its mean log2 expression as beta 0 plus beta j. It's really just linear regression. And to fit lemma, you need to first use this model.matrix function to create a design matrix. So this is an example of a design matrix. You should have, because you have 60 samples, you should have 60 rows here, and 2 columns. One column is the intercept, which is 1, always 1 for each sample, and another column corresponding to the covariate value xi, or specifically in this case subtype 2, indicating whether the sample belongs to subtype 2, like 0 for no and 1 for yes. And you use this model matrix, just like linear regression, to feed the lemma model. And similarly, like DSIG2, you will get a result table. And you use the cutoff shown here, FDR smaller than 0.05 and absolute log4 change over 1.5 to call differentially expressed genes. And for question 3, we are asking you to do pretty much the same thing, but it's just differential methylation. So we will provide you with similar to an expression matrix, this time a methylation data set. You should basically do the same thing as question 2, but a few things to know here is that not all samples have methylation data. So you have fewer samples for question 3. So you can't just use the same model matrix with 60 rows. You need to create a new model matrix, and remember to extract the correct subtype information from your Q1 clustering to build a model matrix for question 3. And another thing is that the methylation data is a fraction from 0 to 1, but lemma takes values from negative infinity to infinity. So you need to first logically transform the methylation data so it ranges from minus infinity to infinity. And you can really just search online how to do a logic transformation. And then you just use lemma as Q2, question 2, to get differentially methylated genes and with the same cutoff. And then the question asks you how many genes are both differentially methylated and differentially expressed. And you just use this function intersect to get the result. And I know this question is kind of tricky. It also asks you like that, is DNA methylation associated with higher or lower expression of this gene, differential genes? So you can really just choose your own approach to answer this question. There is no and absolutely fixed answer. But one possible approach you could consider is like, for each differential gene you get from running this intersect command, you calculate the association between its methylation and its expression. And you'll get a correlation value for each of the gene. And you may check is the majority of the association positive or negative. Another possible approach is for these genes, you just plot their methylation log4 change against their expression log4 change and calculate the association and check if it is positive or negative. And finally, still question 3, it also asks you, how many differentially expressed genes have epigenetic cause? It's kind of vague, I know. And what you should really be doing is check how many among the differential genes you get from the intersect command have opposite differential and the methylation direction. One simple way is you take the product of their methylation log flow change and their expression log flow change and see if you get negative value or positive value, and you take you count the number of negative values. So a unique challenge in survival analysis, actually a unique feature in survival analysis is that survival time is subject to censoring. So for example, we are interested in time to death under a drug treatment versus a placebo group. And we recruited a lot of test subjects, and our study lasts for 2 years. And in the process of follow-up, some people may choose to quit the study because we can't force them to stay in the study. So their data will be censored. That is, we don't know whether they will develop the event of interest like death or not. Like, I mean, they could have developed the event of interest had we followed them for a longer time, but we don't know that. And also at the end of the study, I mean, 2 years, maybe you run out of funding, so you have to stop the study. And so people, there could be some people who haven't developed the event of interest till the deadline, the end of your study. So their data will also be censored. Here are a few notations we'll be using in survival analysis. First is TI, which represents the time to event for individual I, and CI is the corresponding censoring time. And in survival analysis, we really just observe the minimum of these two variables, either time to event or the censoring time, depending on whether the individual develops the event or not. It is usually recorded as a vital status indicator. Yeah, sorry, maybe I was cut off for a few seconds. Remind me, where should I start? Hello?
You're talking about survival analysis?
Yeah, I was cut off for a few seconds, and yeah, okay. We are interested in survival analysis. We are interested in the association between a person's survival given a set of covariates. Covariate xi, which is recorded for each individual, and the survival function st small t is defined as the probability of having the event after t or not having the event until time small t. So in question 4, you will be first asked to plot a survival curve called Kaplan-Meier curve, I think comparing the two subtypes. We will provide you with the survival data, but before you do any analysis, be careful that the sample names for the survival data is different from the sample names in the expression data, because later you have to concatenate and merge two data sets yourself. So be aware that you need to first use stream processing commands to change the sample names so they are consistent across the two data sets. So to plot Kaplan-Meier curve, we need to use the R survival package. And given this data set, you can really just use this command serve to first create a survival object. So yi is the days to death, and delta I is vital status. And remember to move the value of days last follow-up 2 days to death if there is an A in days to death. This indicates censoring, as I just talked about. So, and then you feed the survival curve using the serve feed function. And then this is really just like the linear model. The outcome is a survival object. And the covariate should be the indicator for cancer subtype. And the data is your data frame here. That's why I asked you to change the sample names, because you need to create concatenate a new column yourself, indicating the subtype for each sample here. And then after you get this model, you just input it into ggservplot, and it will plot out the Kaplan-Meier curve for you. And here is an example of the Kaplan-Meier curve. I did not use the homework data, but I use lung disease data provided in the survival package, and it shows the time to death due to lung disease comparing the two genders. I think sex equal to 1 means male, and 0 stands for female. And we can see that males have higher survival probability across time compared to female. But to rigorously check if they have different survival function, you need to use this log rank test to determine if the survival curves are significantly different across the two levels of the covariate. And you should consult the documentation for ggservplot. And there's an option, very convenient. And if you set it to be true, it will perform the log rank test for you and print out the p-value. And the significant p-value suggests, as in this case, like the survival function is significantly different between males and females. And for question 5, we move to another popular area of survival analysis, which is Cox regression. So Cox regression model focus on the hazard ratio against a set of covariates. So we are not interested in the survival function anymore, but we are interested in the hazard ratio. So hazard function at a time t is defined as the instantaneous rate of event at time t, given no event has happened prior to this time small t. So mathematically, you need to calculate the conditional probability is that given that there has been no event until small t, the probability of having an event with a small time period, delta, after this time t. And you divided that conditional probability by delta, and you take the limit, like when delta is infinitely small. So you will get the instantaneous rate of having an event at a small time t, given that the individual has not developed event prior to that. And the Cox hazard model, or popularly, commonly called proportional hazard model, is shown here. So the hazard of an individual at time ti is equal to the baseline hazard times this chunk it's like a linear model and then take an exponentiate. So the thing is like the lambda 0 here is the baseline hazard, which is like all the covariates are when all the covariates are equal to 0. That is the hazard. And hazard ratio is like you just move the beta 0 to the left side of the equation, and you will probably notice that this hazard ratio is irrelevant to time t. It is not a function of t. And this is the basic assumption of Cox regression model and why it's called proportional hazard model. It is that the hazard ratio at any time will be independent of the time itself, but only relevant to the covariate level of individuals. And with a bit of linear algebra, just like linear regression, we know that the exponentiate of a coefficient like beta I is the hazard ratio for one unit increase in the covariate xi if xi is continuous, or it is a hazard ratio comparing two levels of xi if x is binary. And the way to fit a Cox regression model in R is shown below. This is just an example, still using the lung disease data provided in the survival package. We are interested in the association between hazard and sex. So we, like drawing Kaplan-Meier curve, we need to first create the survival object, use it as outcome, and sex is a covariate. The command we will be using is Cox-PH, so Cox proportional model, and input the data. And we use summary statistic to obtain the model statistic. Like sex here, it will tell you whether sex is a significant predictor. It will give you the coefficient, but it's very nice that it will also exponentiate for you. So we get 0.588 here. And this, according to what I said here, this means that the hazard ratio of deaths due to lung disease comparing male to female is just 58.8%. It means males have a much lower hazard of death compared to female. And this really aligns well with the survival curve here. So you can see that although they measure different functions as outcome, the result should be consistent. If you get like a higher survival for male, but a much higher hazard for male, you should check your result to see if something is wrong. And I think this question will ask you about the predictive power of a Cox model compared to a null model. It's also shown in the summary results. So at the bottom, you see 3 tests: likelihood ratio test, wall test, and score test or log rank test. They tell you whether your model has significantly higher power compared to a null model without any covariates. So without any of these covariates, it's just like in linear model, you predict the sample mean for everybody. So a significant p-value, as in this case, indicate that this model is better than a model without any coefficient. So much for the background and how to run the command. So specifically for this question 5, we ask you to use differential genes you got from previous questions comparing the 2 subtypes as covariates to feed a Cox proportional hazard model. You need to rearrange the data to the following format. So you have sample and rows, a column for time and column for death. And these 2 columns will be used to generate the survival object. And then you need to extract the gene expression values for all those differential genes and concatenate them to the survival data frame. But you will soon notice the problem that there are too many genes compared to sample size. I think you have like 60 sample size, like 60 tumor samples, but you could have like 70, 80, or even 100 differentially expressed genes. So you definitely need to shrink the number of genes used to build Cox regression model. And recall machine learning lectures and homework 2 that Lasso can be used as a variable selection method. So you need to first run Lasso on this data set using the following command. You create a covariate matrix. So a matrix excluding any outcome, just a covariate, and survival object, family set to Cox, and alpha set to 1. So this is a panelized Cox model. And just like in homework 2, for Lasso model, you get the non-zero coefficients, and you use those genes with non-zero coefficients to fit the Cox regression model uh, here, like using Cox PH. And then you use summary to look at whether like subtype is a significant predictor, and if the model has significant predictive power based on the results from these 3 tests. And this is the last question for part 2. Question 6, it asks you like, because for many available literatures, using differential gene signatures do not necessarily provide better predictive performance than random genes. This is kind of sad. So the question 6 really asks you, do your differential gene signatures perform better than using some random genes in the Cox model? So you should randomly select a few genes 100 times and make sure that the gene number you select is equal to that of the model you got in question 5, so that the results are comparable. For example, in the end, for question 5, you got 5 genes with non-zero coefficient. And then for question 6, you should always randomly select 5 genes. You do this 100 times, and you fit 100 different Cox models based on random genes, and you check whether any of them could perform even better than your model based on differential genes. The best way really to compare model predictive performance is to use cross-validation, but as you can see, it is very computationally heavy for this problem and not straightforward, because it's not a classification problem anymore. Your outcome is some kind of hazard. Maybe you are not even familiar with how to calculate that. So it's kind of hard to do cross-validation as we did when training like a cell model, like a ridge regression, logistic model. But if you know how to do this, you're welcome to try for better practice, because cross-validation directly estimates the test predictive error. So it's the ideal method. But in this case, you could consider other model statistics such as AIC, BIC, or adjusted R squares to assess model fit. I recommend you to use AIC or BIC. So AIC and BIC are model statistics that contain 2 parts. One part is a summary statistic for your training performance. But as you can, if you take a machine learning course, you will know that a very complex and a very flexible model may fit the training data well, but it captures too much noise and cannot generalize well to a test data. So AIC and BIC also adds a penalty of model complexity and in their formula. So it aims to strike a balance between your training performance and model complexity. So you can't have a really complex model, and it will not generalize well in test prediction. So specifically, AIC is calculated as -2 log training likelihood plus 2 times K, where K is the number of predictors you use. As you can see, the more predictors you use in the model, the heavier penalty you will get. And BIC even penalizes the model predictors more heavier. It use -2 log likelihood plus k times log n, where n is the sample size. And you can see that when n is, I think, when you have more than 7 or 8 sample size, log n is bigger than 2. So this is a heavier penalty. And AIC is more, compared to BIC, more generous in including more variables. So you can, when you feed the model, calculate, record the AIC or BIC, your choice for each model. There's already command for calculating AIC and BIC in R. And you just compare whether for any of the model random gene models could generate a lower AIC or BIC compared to that of question 5. And this is a way to answer this question. So I think this is the end of this lab. I will continue to talk about part 3 to part 4 next week.